So I will try to give you an introduction to the genomic landscape of neuroblastoma, what's about, ongoing, but I'll also go through the basics. So it's really basics. So for the experienced people, might be a bit boring, but they can do a nice morning nap. For the other people, they have to pay attention because we've got a lot of points that we have to go to. Don't read this, but every bullet is a separate subject which we have to go to. So I'll speed up 80 slides in 30 minutes. Let's start. So to start with the basics, what is cancer? Cancer is, of course, fast-growing cells, but in essence, I think cancer is based on genomic aberrations. In every human, there's about 70 billion cells, and each cell's got 1.8 meter DNA folded up into this tiny little nucleus, and that DNA contains information for 30,000 genes, and if there's aberrations in those genes, your cells deregulate, first hit, second hit, and finally you got a full-blown tumor cell. And there's two types of genomic aberrations, in my opinion, where we have the next slide, where you have genomic aberrations, so really true copy number, mutations, structural variations, and the epigenome, so aberrations in the structures that support the genome. In this talk, I will focus on the genomic aberrations, which in my opinion can be subdivided very simply. You can have too much of some parts, so there can be gains or amplifications. You can have too little, so larger regions, loss of heterozygosity or whole chromosomes, or smaller deletions affecting only a few genes. And you can have changes, translocations, and mutations. So these changes lead to aberrant messenger RNA expression, more or less changed, aberrant protein, and then finally functional pathways that deregulate. And then you have a full-blown cancer cell. They are shown by the hallmarks of cancer. So that's cancer. And all these genomic aberrations can be identified using one single assay, which we now have and everybody's happily doing. Whole genome sequencing, main technique now is the Illumina platform that's used. And we can identify all these genomic aberrations in one run. And that then can be shown, that generates a huge amount of data, about a terabyte of data. You can show that in one slide, in one figure where you have all these genomic aberrations, where we put this in a circus plot, where we have all the chromosomes aligned in a circle, and then we can indicate which regions are lost, gained, so the copy number aberrations here in this circle. We can also indicate which translocations occur, so this is attached to that part, that's attached to that part, and we can indicate the mutations, so these are the mutated genes in this specific tumor. So very easily identified and shown, and you can see that there's several genomic aberrations. For instance, when we start on the top here, we can see the gain here. And this is just for this tumor. When we want to show this for a large amount of tumors, we could better unfold this and then show a whole series of tumors. We have about 88 neuroblastoma tumors here, all the chromosomes again, and the copy number variations are shown here. And a gain is shown as red, and a loss is shown as blue. And when you look through the hairs of your eyes, you can see actually this is what's driving neuroblastoma. This is what's driving neuroblastoma. So these copy number aberrations are, in my opinion, the main drivers. So the first and most common aberration that you can see is, of course, the gain of chromosome 17 or 17q. Some tumors have gain of the complete chromosome. Some tumors have gain of only part of the chromosomes. So 17q occurring very frequently in neuroblastoma. A few tumors not, but the majority do have. And the current idea is that this leads to a copy number gain of a common region, which is shown here. So this is the region that is frequently gained. And the paradigm is that this frequently gained region contains several genes that are tumor driving, that support neuroblastoma, neuroblast deregulation into tumor cells. So it's not one gene, but it's several genes. And the idea is that these genes are located in this frequently gained region. So this is the average gain shown in neuroblastoma. So there's some tumors have complete chromosomes. There's tumors with breakpoints in this region, and then most tumors have this region gained. So frequently gained region. And there's all kinds of genes that are located in this frequently gained region that are of interest. For instance, on the tip, we've got BIRC5, which is survivin. a clear inhibitor of apoptosis. Protein interferes with apoptotic pathway at several locations, so thereby tumor driving. But we also have the NME gene, which was described several times to be a transcriptional target of NMIC. So you've got the NME located on 17q, there's NMIC, and then there's even more NME, so that's usually overexpressed, and that then inhibits CDC42, which is actually involved in neural differentiation processes and cell growth. So other examples will appear during this meeting. So for instance, we have the PPM1D, which is going to be presented tomorrow in parallel too by Jelenka Milosevic, which is with one interesting gene involved in PVV3 pathway. We got several genes presented by the group of Frank Speleman. The Custer is showing on the parallel session about TBX2 and BRIP1, and there's more candidate genes. So many genes located on 17q. This is 17q. We have other gains. Other frequently gained gene, which is not that frequently worked on, is chromosome 7, frequent whole chromosome gain. Interesting region. So if you want to work on an interesting gene, try to find one on chromosome 7. There's a lot of potential tumor-driving genes. Other genomic aberrations, so 2p gain also occurring, and that involves, of course, the NMIC gene and the ALK gene, which are known tumor drivers, but mainly deregulated by different aberrations, which is, of course, for the NMIC gene, the amplification, where we have here a circus plot again, the chromosome 2 gain, but you can see that there's something happening, and there's a small black dot there. And when we focus on that region, we can see that this is a high-level amplification. So this is copy number again, and then we have here a log-fold changing copy number, and this is, I think it's about 7, so that's 0 is that my time? No. Oh, 7 minutes left. So we got 0 is 2 copies, so that's 4, 8, 16, 32, 64, 128, 256, more than 256 copies. So a huge amount of copies you have of NMIC there. So this is really striking. So how does this look? Well, we know, of course, the FISH analysis, fluorescent in situ hybridization, here with an NMIC probe, and we can see here this homolog staining region. So you get all these copies of NMIC fused to each other and fused to one chromosome. But this is the homolog staining region, which occurs mainly in cell lines and in some tumors in older children. I thought this was also happening in other tumors, but apparently this mainly looks like this, which is the double-minded. So these are small circular parts of the amplicon which are scattered around in the nucleus, and they multiply each division, and then they stick to the chromosomes, but randomly, and then are subdivided over 2 new cells. And you can imagine that is a hugely powerful mechanism to extremely fast amplify the number of NMIC copies that you have. So, and this is how it actually looks. And of course, we know MICAN, that is the transcription factor together with MAX, drives a huge amount of genes that cause deregulation of many different pathways that are involved in oncogenesis. And there's a whole session on that, the parallel 7 on the Saturday. So that's a very interesting session. Also, potential new therapeutic interventions. So this is all too much. We can also have I wanted to show a few more amplifications. So we've got CDK4 amplification, but these are all rare. MICAN occurs 25%, CDK4, cyclin D1, Lin28B, so these are genes involved in G1 regulation. This is that 7 pathway, less frequent percentage or less. So this is all too much, but you can also have too little. We know the chromosome 1p deletions, which occur most frequently together with amplification of NMIC. The 11q deletion associated with poor prognosis. And in the early days, the idea was that these regions were deleted, and it was actually a 2-hit model in which the classic 2-hit model in adult cancers functions by that you have a paternal and a maternal allele. For instance, APC, you get one mutation, and then the other allele is deleted, and then you have a complete inactivation of this strong tumor suppressor gene. So then you have a cancer cell. But this is actually not happening in neuroblastoma. We don't see the two-head model. We could never identify these mutated genes on 1p36. Everybody been sequencing genes there to identify those two-hit genes. Never found it. 11q the same. Well, maybe somebody will show during this meeting, but I don't think so. I think the paradigm still is actually the same as with the gain genes, that you have these frequently lost regions. So for instance, chromosome 1p, so this is the average loss. So there's gain sometimes of 1q, but loss of 1p, and this is the most frequently lost region, 1p36. And that contains then several tumor suppressor genes like CHD5. There's the MIR34A involved, ARID1A, which is involved in SNF signaling. So it's really various genes that are located in that region. And then the idea is that these genes are missing one copy, and thereby there's haploinsufficiency, so reduced expression. You still need some expression of the genes, but there's reduced expression that finally leads to oncogenesis. So that's the idea. You can have large regions, but there's also small deletions. And these small deletions can affect single genes. And there's actually one small deletion which occurs quite frequently. You can't see it, but it's located here on chromosome X. You can see there's one copy lost of chromosome X, and there's something small happening there. And when we focus on that region, we see that these small deletions occur, and they occur in several tumors, and they occur in the ATRX gene. And thereby they inactivate the ATRX gene. And this causes a reduced expression. So if we look at the messenger RNA expression in neuroblastoma tumors, and these three tumors are the three tumors that have genomic aberrations, and they all have a very low expression of ATRX, and that then apparently leads to alternative lengthening of telomeres. So huge telomeres, which is one of the hallmarks of cancer, to maintain internal growth. And you can see here, this is in other neuroblastoma, and this is the ATRX mutant neuroblastoma with huge telomeres. So those are the lesions. Then we can have changed DNA, translocations. Well, classically, we know many translocations. The translocation is, of course, the chromosome 922 translocation, which creates a new fusion gene, BCR-Abelson, against which we had the therapeutic intervention, which was so successful. But then this also does not occur in neuroblastoma. We don't get these fusion genes. In pediatric cancer, we do get in solid tumors as well, PAX4-CAT, EWS-FLI, leukemia is a lot, but neuroblastoma not. Well, very rarely. Very rarely. Yeah. But what we do see is translocations. And what we do see translocations, and these translocations have been found to indeed be tumor driving as well. Well, we have one happening here from chromosome 11 to chromosome 5. And we see in about 10% of neuroblastoma translocations that finally end up to chromosome 5. It was first unclear what happened. There's actually nothing happening to the gene, but the translocation occurs most frequently right in front of TERT. And in front of TERT, it's a epigenomic desert. There's nothing happening there. But then what happens is that there's a translocation of a very active enhancer region. You can see here, this is the chromosome 11 region, very active enhancer here by the histone modification. And you can see indeed this is right attached in front of the TERT gene. And thereby you get an upregulation of TERT and upregulation of telomere lengthening. So those are the translocations, most important translocations. And then we have the mutations. And that's also very poor in neuroblastoma, because we have a paucity of mutations in neuroblastoma. When we filter for the mutations that are in the coding sequence, that are changing, that are somatic, we end up with this figure. So these are all neuroblastoma tumors. This is the frequency of mutations in the coding sequence that are somatic. And that, on average, is 12 mutations per patient, which is really little. Also, if you compare that to other tumors, so these are adult tumors, mutated phenotype tumors, these are the pediatric tumors. So in general, neuroblastoma, specifically in pediatric cancer, have very little mutations. Well, it wouldn't be a problem if these were all strong tumor-driving mutations, but when we look at the results from the Pugh paper, the group of John Maris, we see in this quadrant the mutations in all the different tumors, and all these tumors have nothing that drives them. We've got ATRX here, which we just talked about, but those are structure variations, and the only mutation which occurs quite frequently, about 9 to 10% in neuroblastoma, is the mutation in ALK, which we know, and these mutations occur in the active domain, kinase domain of ALK, and there's two hotspots, the 1275 and the 1174, that are mutated most frequently. There's other mutations occurring as well with low frequency. A very nice paper by Jael, Mosse, which indicates all these, aligns all these mutations, because not all are transforming. So beware, but you can find them back in that paper. Then there's one mutation which was not shown in the previous figure, but I still want to call it the forgotten mutation. So we have the FOX2B, which is also quite frequently mutated, about 3% of neuroblastoma, which is significant, but it never shows up. It's actually mutations that occur mainly in this alanine-rich region, and thereby cause a change of the gene, most likely inactivating, might be dominant negative. But this is not occurring in the current studies, because if you look at the coverage when you analyze by whole genome sequencing, this is the same gene turned around. So this part is the part where the mutations occur, and this is the coverage in all these different genomes, and yellow indicates no coverage. So it's just a very difficult region to sequence. So beware when you analyze the data, when you use whole genome data, that this is mostly not covered, and it could be that there's still mutations occurring there. So these were all the genomic aberrations, and of course these genomic aberrations can be used for various purposes, also for classification, identification of new therapeutic options. I want to go briefly through the classification, but Gudrun will also do that. So classically, the INRG system, which is now most commonly used in the world, where we have the localized, the metastasized, and the formerly known as 4S tumors, where NMIC is used as a biomarker for poor prognosis and high risk, specifically in the localized disease and also in the younger children with metastasized disease. And 11q is used as a biomarker for poor prognosis, also in several subgroups. So this is how it's currently used, and you can find it. I think there's changes coming. So as I said, when you look at this schedule, these are the copy number aberrations again. And they're clustered based on the type of or the copy number pattern that occurs. And you can see that there's certain patterns occurring. So these seem to have whole gains and losses, this partial. And when you relate this to the INSS staging, this still is, you can see that these patterns correlate to the INSS stage. And so here this is all high stage tumors, but here's low stage. But you can see there's, for instance, one tumor with a high stage tumor in between of this group. So maybe we could use this data as an indicator, and that was published by the group of Delattre and Speleman, where they proposed a subdivision in 5 subgroups, 3 without and 2 with MICAN amplification. So we have type A, which just have the numerical aberrations. Oh, 9 minutes still. Can slow down. So you got we've got numerical aberrations. And in the second group, we have the segmental aberrations, which occur. Then there's a mixed group that has segmental and numerical aberrations, and then 2 MICAN amplified, 1 without numerical aberrations, and one with numerical aberrations. And this is, of course, what you do see here as well. So these are the numerical aberrations. These are the segmental aberrations. And indeed, it looks as if this correlates with the pattern. So this will be used, and because these groupings really correlate strongly to prognosis. And I think this could be incorporated in the classification system. This is one future, but there are, of course, always other futures, which will be presented this Thursday on the plenary session. I think 2 or 3 talks about that, where we and others have identified the aberrations in telomere lengthening by TERT, ATRX, and then MICAN, which is transcription upregulating TERT. And you can see that these are more or less mutually exclusive, at least TERT and ATRX, MICAN, some overlap. But apparently these are the ways by which you can maintain your telomere lengthening. There's some alts which are not ATRX mutated, but these groups all correlate to a very poor prognosis as shown in these Kaplan-Meier curves. So there will be a debate whether we could use this indication with some additional genomic aberrations for subclassification as well. So those are the classifications. There's, of course, genetic predisposition. You've got 2 kinds of genetic predisposition. So you've got the predisposition where you have a slightly increased risk in a larger population, or you have the familial neuroblastoma. So 2 types of predisposition. Genetic susceptibility, that's a difficult word. best found by GWAS analysis, which is mainly done by the group of John Maris. So genome-wide association studies, you use a large amount of SNP arrays of neuroblastoma patients and compare that to a control population, and then you can see whether certain SNPs are enriched in neuroblastoma. And there's some interesting candidate genes, so BART1, LMO1, recent second study, Lin28B, which we also saw as an amplified gene. So there's several interesting candidates. And if you put this in a plot, it looks like this. So this is the risk of getting neuroblastoma. So the effect size of a mutation to get a neuroblastoma in the end, and this is how frequent these events are. And these events actually occur in the quite frequent range, but they give a slightly increased risk of getting neuroblastoma. But then, of course, on the other hand, we have the aberrations that are very rare, but give a very strong increased risk of getting neuroblastoma. Best known are the ALK and the FOXP2B mutations. So ALK is occurring in 80% of the familial neuroblastoma, and FOXP2B is in 10% of the neuroblastoma. There's some other rare events, but these are real rare that can lead to neuroblastoma. But then there's still a portion, which is about 10% of the patients, that do have familial neuroblastoma, but do not have genomic aberrations in any of these genes. So never tell a parent that it is impossible if there's no ALK or FOXP2B. We had the case, brother and sister both died of stage 4 disease without adding any of these mutations. So there's some exceptions. So these are the genomic predisposition patterns. Now we have some interesting mutational patterns where we have chromatopsis, mutational signatures, which are really hot now, and the silent genomes. So chromatopsis, here in this circus plot, which we call the moustache, nice area here, and when you focus on the moustache, it looks like this, where you have this huge amount of structural variants. And these are all copy number aberration, and you see it varies between two distinct states. So this is chromatopsis, most likely caused by shredding of chromosomes, which are left behind during cell division, get encapsulated in a micronucleus. Then degradation starts, but by coincidence, refuses with the nucleus, gets repaired, and then you get this massive amount of rearrangements when these parts of chromosomes are glued back together again. Two questions. Why can this happen? Well, it happens because these genomic aberrations finally lead to genomic aberrations that are oncogenic or affecting tumor suppressor genes. For instance, here on chromosome 5, we saw that this rearrangement, one of these rearrangements, caused actually translocation of a strong enhancer in front of TERT. But there's also CDK4 amplification, MYC, LOHF1P is mimicked. So really the known genomic aberrations are mimicked by these chromatopsis events and thereby selected for. The second question is, how can this happen? Well, when you do see these chromatopsis events, you have to be aware that there could be something wrong in the germline in DNA repair patterns in these patients. So 2 out of the patients that we have identified having chromatopsis showed de novo germline p53 mutation. So these were de novo Lieframeni patients. So beware when you see a chromatopsis pattern to look for germline aberrations in DNA damage signaling. And actually that's of importance because if you want to irradiate these patients, you should take real care to really consider whether you want to do that or not. One of these patients indeed was cured from his neuroblastoma and then finally died from a glioblastoma, secondary tumor in the brain. So beware when you see this. So mutational signatures started after the publication of Stratton with this paper where he showed that there are certain mutation patterns, and these mutation patterns, so you can have different types of mutations, C2A, G, CT, TA, TC, TG, so these are all the options, and then you can subdivide by the surrounding 2 nucleotides. And then you get these patterns, and there's all different kinds of patterns that you have, most frequently involving C2T mutations. In neuroblastoma, we do see frequently C2A mutations. And, well, still needs to be published, but we think it's got something to do with defects in the 8-oxyguanine signaling, where 8-oxyguanine defects in this pathway lead finally to C2A mutations. And most strikingly, neuroblastoma, which have this pattern in C2A mutations, show defects in OGG1 and MUD-YH. OGG1 is located on 3P, MUD-YH is located on 1P. So these are deleted. haploinsufficiency, increased risk of getting these events. Finally, silent genomes. So there's always story about neuroblastoma silent genomes, no genomic aberrations. I think case, of course, 1 to 10 will be that there's infiltrating stroma, there's missed biopsy, so that's reason 1. But for the sharp person, here on bottom you see that I did include 2 blank genomes. So what happens to those? Because I think those are neuroblastoma, they really look like neuroblasts. And when we analyze these tumors, they look like this, so there's nothing happening, but there's something happening there, and this is real, there's something happening, and that is actually a translocation of the FOXR1 gene. So here's FOXR1, and there's a promoter which is attached to FOXR1. So it gets a new promoter. Normally, the FOXR1 is not expressed, but then by another promoter, FOXR1 is expressed. And this single event is enough to cause neuroblastoma. We go back like when you, so this one event is then the same as having all this. So I still think that studying fork box transcription factors in neuroblastoma is nice. So anybody having time left, go and do that. Then finally, well, this is what the genomic aberrations are, more or less, in neuroblastoma. There's, of course, more things happening, but in general, these are the big things, in my opinion. But that's not stable, because there's a lot of heterogeneity. Not every tumor cell is the same. Every tumor cell is different. And there's tumor evolution. In time, tumors change. And that we have shown in triplets when we go back. This is the tumor before treatment, and this is after treatment and relapse. Oh, wait. And then you can see that there's a huge increase in the number of mutations and the structural variations. So tumor evolution. When we look what type of events are occurring, 10 seconds, we have here primary relapse, chromosome 17q, and there's the NF1 gene being completely inactivated, which causes an activation of the RAS-MAPK signaling. Another event here, 1 MECAN amplification here, a second line appearing, and that is actually the ALK amplification, which is de novo, that also activates the RAS-MAPK pathway. And we do see that in relapsed tumors, there's a high frequency of aberrations in the RAS-MAPK pathway, which is not occurring in the primary tumors. I think this is quite a high percentage, but there's a strong enrichment, and I'll be talking on that on Friday. That's it, 30 seconds late. These are the papers which were used for this presentation, and I'll leave the slide like that for you to make a picture. Okay.